Hello, everybody, and welcome to our class on starting to analyze our custom ChIPseq data. So we've been learning a bunch of fundamental skills and curated a set of DNA binding proteins and ran the NF Core ChIPseq pipeline got the peak files for that collection of proteins. I made my own, you'll have your own, but the same code will work on all of ours. So I just want to point out where we are in GitHub in a second as usual, but just in case you're following along online here, you can go to this website here, linkrna.io, and teaching is where we are here on this tab. And so you can click there to get an overview of the class. But if you're at this point, I just wanted to show you we've done our bash skills, connected to GitHub and cloned the class, run ChIPseq, started to look at those peak files, learning some R basics, and then overlaps, made some custom functions here. So we can now just get our peak files and find peaks that aren't common in all of them and start analyzing that data. And that's where we are now is number 6 here, ChIPseq analyses. If you're going on the web, we have finished this section here where we made 2 custom functions and have now moved on to 6 here on the website, ChIPseq analyses. Okay, and as usual, we're here in the GitHub, on your GitHub now that you've cloned it. And we've finished 04 RNA, our basics, and moving on to our analyses. And if we click there, what we see is all the different types of analyses we're going to do now that we have this data run and the peak files. And so we are going to make heat maps. We're going to cluster them. We're going to make metaplots of where they bind and how it looks. We're going to add RNA-seq into this, and then how binding of ChIP-seq data and RNA-seq intermingle. And we'll do this all sort of stepwise. So that's where we are now, and we are starting with setting up this analysis and starting to peek at it with 00 consensus peaks, and then 00 create consensus peaks. And then I will be running that same code in my console here on RStudio. Okay, so let's get set up. We need to source our import peaks and create consensus peaks functions that we created previously, and load in genomic ranges, tidy range, verse, etc. So let's load these and source our function. So now in our environment, we have our import peaks and our count peaks, or our consensus peaks and our fun peaks from before. So, and we're going to make more as we go and populate this later. Okay, so now what we're going to do is use import peaks. And to do that, we need to set the folder path. This can be different for you if you're at home, but in class, we're just setting up a base path to your folder in class. That way we don't write over stuff. Then we go to where the peaks are in our O3 next flow. In classes O3 next flow, you ran your 00 my ChIPseq folder, and these are where the broad peak files are. And our import peaks function also will search for the .broad peak extension. Okay, so I'm going to go back to my working directory here, and 00 consensus peaks for the start of our analyses. Okay, so let's get to this data. And I have a base path, make sure I have peak path, broad peak path, and I'm now merging those two to go right to my peak files with the file path function. So it's in a readable way that the system will then go right to that directory. Okay, so step 1, because we have this handy import peaks function, we need to tell it where that file path is, and I just made broad peak file path to do that. So we can make that peak list, and it's going to take a minute to have it import those peaks as a list of G ranges that has a name of the DNA binding protein that's associated with. looks like this. So we can see this is a protein name and the replicate number next to it. And we can keep going. So that took very little time compared to making the function. And so now we are going to take each of those, we're going to find the number of peaks in each file. And so we're going to do sapply over peak list, which has all of our replicates in it. And we're going to do the function length, and we're going to pipe that to a data frame. So let's run that real quick. So I'm going to go through each replicate, find the length of the number of peaks that are in it, and pipe that out to a data frame. Okay, so let's take a look here at peak num. We can see we have weird column names. We'll fix this in a second, but each replicate and the number of peaks that there are, and we can sort this by lowest to highest, etc. Okay, so we are just collecting a little bit of information. We're gonna have our first result, just how many peaks are there. We quickly imported them, and now we can run a little bit of analysis on them with applying the length function, which will tell us the length of a given ChIPseq file. And we'll now label the names of that object as num peaks. And so now if we look at peak num, we can see that this is num peaks, and this is the DNA binding protein and replicate number. Okay, so now we are going to make a DNA binding protein column. We're going to make a third column in peak num here. We're going to split this replicate number and keep the replicate number and the DNA binding protein name separately. So we can do row names to column and make a new column called DBP. And what that's going to then pipe to doing is a function of separate the DBP column into DBP and replicate and separate by the underscore. And that underscore will be right here. And so it'll separate the two. This is an R function, separate. And it's going to take this character string and separate them into two different columns. And the one column will be named DBP and the other column will be named replicate. So that's what separate function is doing here. Its parameter is which column is DBP, which has been piped into it from up here. Separate DBP and replicate is the new column names, and separate by the underscore in there. Okay, so if we do that, we can write it out and look at peak num. And we see a pretty cool little data frame that has a bunch of information for us. Our replicate number we can sort on now, or grep on, and number of peaks and DBPs. We could plot a histogram and facet or something by the DBP name. So there's a lot we can do with just this little bit of information, but we have our first data. So let's write it out with a function write CSV, which needs the parameters of the peak num data frame, or what to write out as a CSV. And we're going to give it peak num, which is this, and name the file. And it's in results here. I've made a results folder so we can start to collect results in this analysis directory and write it out. And now I can see in results I have num peak df, and I can view the file and see the same data, just now with commas separating it. Okay, so a little bit of results on our thing, and now we are going to run create consensus peaks to find a consensus from reduced function we made, and we are going to run it on this peak list of all of the DNA binding protein data you selected. We're going to sort. We're going to go in and find for each DNA binding, unique DNA binding protein, all the replicates, find the peaks that are in all the replicates, and then export that, or keep that as a list for that DNA binding protein. And we can see how that looks in a second. And there's a few ways you could do this. You could just type in the names of your DBPs here. Or we can get that from names peak list that we just imported, and we can see the names of each of the peak files here. and then we could type in paste in the unique name, but that would take forever for 480 DNA binding proteins, which we're going to do after we practice these. We're going to do it on a much bigger data set and rehash over these same exercises. Okay, so then what we could use is our DBPs function, which is part of our create consensus peaks function. We need, when we built that function, we use this as the input into the function, which is a list of unique DNA binding proteins. So now we can take the names peak list and take all of these names here, assign them to X, and then string split X by underscore and take the left object, which is the name of the DNA binding protein. And so if we just run that over each of the names in peak list and string split them by underscore and take the left side or the first value. Okay, so now we get an object DBPs and we can take a quick look at it. And now we have a unique DNA binding protein to put into the consensus from reduced function. And so we can run a for loop saying for each of these DNA binding protein names, but we could also just use an lapply here where we say for lapply over each of these names, do consensus from reduced function, and then the object to do that from is peak list. So it's basically a for loop for I in DBPs, do run consensus from reduced, and using peak list, which is created within consensus from reduced, or sorry, our peak list of all of our objects here, which will individually be selected, and the peaks that are in all of them will be found. Okay, so just one more time, we're going to take a for loop, essentially lapply for each of these DBP names and grab all the replicates, find the peaks that are the same in all of the replicates, and in this peak list is where the lapply function will occur. Okay, so let's run that. And we are now going to take 7 different DNA binding proteins, each with multiple replicates. And for each of them, find the merge of all of them. And it took just that long. That was not bad. And so let's take a look at what we got. Consensus lists. Oh, it's just a numbered list. So which one's which? Well, luckily we made that DBPs object here, and we can make the names of the consensus list that DBPs, because that was the same object that went into our function here. Okay, so let's try that and see what happens. And now we can see consensus list here is a G range of the peaks that starts and stops of peaks that are in all the replicates. And there's a list of those peaks for each of the different DNA binding proteins. And so we've reduced a larger set of data into something we can move forward with and say, these are the peaks that are very consistent and reliable in the ChIP data I have. Where do they land in the genome? What types of genes and places do they land? And that's what we're going to do next. So we're generating the data we're going to use moving forward. Okay, so cool. That's really handy. And later we're going to print all this stuff out and organize it. But for now, we're just doing the concepts, and we'll go through it again and learn through some iterative processes. Okay, so now we can look at the number of consensus peaks. We previously made that num peaks data frame, that num, what we call peak num object that has the replicate and the thing. Now we can figure out how many peaks there are in the consensus list versus how many there were in each replicate. And so there we're using this sort of built-in for loop sapply here, where we're going to go over our consensus list, which is now just the single DBPs, common peaks that are common in all replicates, and apply the function length. So we did that same kind of thing for each of the replicates in peak list instead of consensus list here. Okay, so then we're going to make that a data frame, and we're going to name the row DBP, and then we're going to rename it. But I'll just run this part here so we can see what's happening. So we're going to get a num consensus peak data frame. And we see that there's the name of the DBP consensus peaks and the number of them here. Okay, so now all this is doing is renaming the column to DBP instead of dot, or sorry, instead of nothing. And that's what row names to column as a function. And it's going to take the first column and label it DBP here. Run that, and now look at our consensus peaks. We can see that the column name is now DBP. We still have this dot here. We might want to replace that and say use dplyr, which is part of a great way to rename things. So dplyr : rename, and then do what object are we going to do it on as number of consensus peaks, and change the dot to number of consensus peaks. So it's kind of counterintuitive order in my opinion, but it's renaming the new name from the old name here. So now we can just run this whole little bit and look at our num peaks here and see that we don't have a dot here, and have another nice data frame that we can merge in with our peak files, or just cross compare. How many peaks did we lose when we looked for consistent peaks across replicates? And so we can join that, do a left join with peak num, which we made before, which is the DBP replicate and the number of peaks in each individual file. Things open again. And then so we will merge these two and take a look at what happens there with the left join function. They each had a DBP column with the name of the DNA binding protein, and merge them. And if we look at peak num now, we can see the DNA binding protein number, the replicate number, and since it's the same, there's only one value for the consensus, so it just filled it in each of the columns for the given DNA binding protein. So we can see that, going from 79,000 to 7,000 seems like a lot, but some of them are 14,000 going to 7,000, which isn't too bad. So something to look at. It's a good way to QC your data, and that's what we're in the process of doing. Is if we just got fresh data, we want to look at get reliable peaks and see how they changed. Okay, so we can now write that out as well. We have this handy data frame with information on our original peak numbers, and now our consensus peak numbers. Okay, so let's export these files. We've always been at this point here where they're in our environment here. We have peak lists, we have all these great G ranges named in a list here. But let's export them now at the consensus peaks that we have here. Each of these is a G range, so we can use rtrack layer export and export them out. And so I'm just making a path to where I want to output these, and I'm doing that here in my working directory, OO consensus peaks in the class O5R analyses. I don't have this analysis anymore since this has been updated, and I'm in 00 consensus peaks, and I'm going to export it to consensus peak as a folder I made here. You can make a new folder called consensus peaks there. So we're going to share this path, but this is the path to your specific folder where you cloned the class originally, and I think I already have it in there as the way I want it. So then this is where I'm exporting them, is from the cloned classes 05, 00 consensus peaks, and into a folder called consensus peaks. Okay, so I'm going to make that file path and then merge the two with file.path function to import in. And so that's my export path now. That's going to be a variable used here for where to rtrack layer should export them. So normally what rtrack layer just needs is the name of the object and the environment, and the file path of where to go to. So we're going to need to run a for loop because we have multiple files in there. Another good example of using a for loop to make work easier. So now what we're going to do is, if we look at consensus list here, we have 7 entries of G ranges. And what we're going to do is, for I and length through those 7, we're going to run track layer export. And now we need to tell it where to export it to, or what we're going to export is consensus I, the first one, so EP300 in this list. And then the next part of our track layer parameters are what file, and then where to put it. And so where we're going to put it is export path, which we just set up here to where you want it to go. And then it's also going to paste the names of consensus lists, which is the unique name of the DNA binding protein of I, which again, in this case is EP300. So this is just running that names function we've done several times at this point for the entry that's currently going through this for loop, and then also paste on the text _ consensus peaks.bed. So this is going to export them and name them, and it's going to take the export path and then add on the name of the DBP that is being currently exported, and then append this text to it. So let's run that and see what we get here. And it's going to take a second for it to export it. But if we look now in consensus peaks, did I not do the right export path? Because I did change this. That did not work. Okay, sorry about that. I had a file path error. I've troubleshot it, and I'm back on here. So the key thing is, if you have file path disconnections, it's very common. I changed my consensus path. Long story short, I'm running back the same code to make an export path for the for loop we're going to run, where we're going to take the length through consensus list, which is each of our consensus peak files as G ranges, our track layer, export them, the consensus list I that it's doing, and paste the export path and the names of the DNA binding protein that's being investigated here, EP300 again, and then paste on this text here. And so now we can get this to run, and here we populate them in this folder here. So what we can see now is we have a bed file that we can now use to go visualize data. That's what we're going to do soon, is take our exported data from the NF Core ChIPseq pipeline, put it into viewing browsers such as UCSC Genome or IGV, and then we can import these peak files, these .bed files, and look at where our consensus peaks are versus individual peak files, etc. So now we have those in a handy format that we can use later, and they're already named for us based on, if you want to go back over what we pasted in here in the export. If you were doing this locally, you could just paste the place you want it to go directly in your folder. That's because it knows your working directory. So you can have it in results, consensus peaks, etc. The same thing would happen. And so now let's take a look at what our track layer export did real quick before we just move on. What did we just get? What do these bed files sort of look like? So I'm going to read one in with read TSV, which we've done before in our basics class, and read in this EP300 thing. Again, we went over why call names is false. So we don't want our first row of data becoming our column names. Okay, so let's take a look here. Peak 1. Am I in the right working directory? No. Okay, sorry, I'm having file path issues today. This is common, as you can see. I'm having it right now. So I just put in the full path to where I want to list the files from, and that's where I'm working here. You can get that from right here, copy folder path to clipboard. And I just sent it straight there. Full names is true for the path, and the pattern I'm looking for is .bed this time. When we've run this before, we've used pattern equals .broadpeak, and now they are called .bed files. So we're going to list all the file names that have .bed on them from this directory into an object called consensus list here. And if we look at consensus list, it's the long file path to the files. We can look at it here as well. And we can see that I'm going to the rest consensus peaks or H3K4 consensus peaks are now the file paths are in that object. So now we can do an lapply or like a for loop over that object. And so basically each of those, for each of the lines in consensus file list, it's going to do read.table and read that file in and change the column names to what they're supposed to be, which is chromosome, start, end, name, score, and strand. That's the broad peak standard nomenclature outlined up here. And so let's do that, where we're going to do an lapply, which is like a for loop over consensus file list. So for each of the files in this 7 lined object, it's going to do read.table, read it in, and change the column names to this. And now we're going to be calling this peaks instead of, and we'll just call this peaks for now. Okay, cool. So let's put the names of peaks as DBP. We can see it's a list of 7. This is our 7 DNA binding proteins that we're looking at. And we're looking at their loading in their consensus files. And so now let's name them by putting in our DBP name. And we can see peaks now has the name of the DBP associated with it. So that names function is pretty handy. And these are the objects that created the names of peaks. So we know they're in the same order. Okay, so now the last thing we want to do is get rid of these non-canonical chromosomes. There's a bunch of contigs in here. It gave us all these warning messages that will be annoying to fill up our screens later. So let's filter out anything that's not in a normal chromosome name. So we have here paste 0, chromosome 1 through 22. So we'll get chromosome 1, chromosome 2, chromosome 3 through the 22 numbers being called for it to repeat this. And then chromosome M, X, and Y. And so then we'll have a string of chromosome 1, 2, chromosome 2, chromosome 3, chromosome 4, chromosome M, chromosome X, Y, and have that as an object. Okay, so then we can see our canonical chromosomes is here. We have chromosomes 1. So this number was just pasted on 22 times. In this paste 0 function, and then concatenated with these as well. Okay, so now we can go do an lapply across our peaks object, which is now named list of G ranges for all of our peaks. Each one of those has some of these non-canonical chromosome contigs in it. And so we're going to go through with an lapply and go through each one and intersect out anything that doesn't match these names that we want in our data. Okay, so we're going to use the lapply to do that across peaks, and we're going to do function X. Here's a fun one, good tidyverse usage here, is we're going to call up a generic function X, which we're going to use to pipe into this bit, but we're going to pipe X from function X, which is now each line in peaks, and we know each line is like going to be EP300 and so on as it goes down. Okay, so for each of the lines in peaks, it's going to run function X, push X into filter, and that's filter chromosome in canonical chromosome. So we just made this canonical chromosome thing here, and in all the peak files, they'll start with CHR. So we're filtering only things that match these strings here. Okay, so we're going through each of the G ranges peak files as X, and we're running filter of has the word chromosome in canonical chromosome in the each peak file. Okay, so let's run that. And now those nasty chromosomes are gone. And that will help us later. And so we have a nice clean set of peaks that are reproducible in all of our replicates and don't have any weird contigs that can throw off programs down the road. So this isn't as esoteric as it seems. It's important to usually get rid of non-canonical chromosomes that can cause errors in other programs. Okay, so now that we have these all cleaned up, let's print them out. And so to do that, we're going to make an object called new files name, where we're going to paste consensus peaks as a / names of the peaks is going to be the name of the DBP here, and underscore consensus peaks.bed. So we can do that, and then we can use this file names as an input into a for loop to place the new file name on each one as it goes through this for loop. So it'll call up new file name, and it'll be calling the names of peak I, which is the DNA binding protein name, and then underscore, adding this underscore bed there. So there's a little bit of function running to make a name of a file is essentially what we're doing here. And I'm going to do this, okay, there. And then, so now we're going to run the for loop over peaks I, which is one of the 7 DNA binding protein peaks G ranges, and do the new file name I, which is now going to do names of peaks I, and then just some tidying up to paste it into to export it into our file here. So it's going to have column names now and other things that we'll want to use in the future, and no chromosomes. So we'll run that there, and voila, we've got new nice clean consensus peaks with that'll have headers now sorry, we'll have none of those non-canonical chromosomes. And anytime we read this in as a bed file, we're going to have to rename the columns as with that little tiny bit of code. But we will not have any of those weird GL chromosomes that we had before. Okay, so that is great. And so now these files can go straight into visualization programs like IGV, which we're going to go over in another video. But if you want to do them on UCSC, there's one more step. Unfortunately, we have to put a header on the peak file so that they can be read in from your computer. We're going to do a video on UCSC and how to take the peaks and upload them. We're not going to re-go over the code in that video. So if you are interested in looking at your peak files that you've just made here and can download to your computer and start looking at them, if you want to do that in UCSC, you need just a little bit more here, and I'll go through that in the next few minutes. And so, but it's not that long, trust me. So, okay, so we have a bunch of information here if you want to read more. And as you do this more and more, you're going to read this stuff more and more. So I'm just giving you a resource of where to look. But it has information on what a broad peak file is and what type of header it's going to need. And I'm just using bed graph here for no reason really, except for that it works. It's a fine header. We're just adding something so when we load these into UCSC, it knows that we're loading a peak file. Okay, so the header, what we need, as you can see, we're setting up something for another way of doing this here. So we have, we're going to take the track, and this is what UCSC wants here. Track type is bed graph, name equals, this is not a bed graph, this is just a bed file. Forgive me. We can look it up more in here of other types of things that could go in this entry. and then the name equals the names of peaks. So then if we run a for loop on this, we can then, for each of these, put the DNA binding protein into the name of the file that we export with the header. Okay, so we're just making an object that is pretty boring, that's just putting this text together and is going to be able to grab text from names peak in it. So let's do that to look at headers. Okay, so now what it did is for each of the names and peaks, it made, it pasted together all that information. Okay, so now for the new file names, we want to paste where we're going to put these, and where we're going to put them is UCSC consensus peaks here. And that is in our working directory already. If I go back to my working directory, we can see UCSC consensus peaks here. And so I'm going to have the new file name exported to UCSC consensus peaks so I can keep track of what type of new files I'm making, the names of the DBP of a given DBP, and .bed on the end of it. Okay, so now we can run a for loop over that new file's name object, and we can see here we just have made where what the output is going to be for the for loop, or sorry, the input into where it's going to go, and then export the file to. Okay, or sorry, the new file name. Okay, so in 1 through length peaks, which is our full G ranges here, we're going to do right lines header and new file name, and then this is going to be that header up here will be called for DBP1, and the new file name that we made here is going to be the new file name of the UCSC peak file. And then we're going to call the function write table for the peak file, consensus peak file we're looking at. We're going to add the new file name to that consensus peak file, separated as a tab, file name, and call names, row names, false, other things that we'll get into more later, but are really just making sure you're just writing out a very plain file. Okay, so let's run that here and have that. And voila, we now have that, and we should have our UCSC consensus peaks here. And if we look at them, we can see now they are the same thing. They've as here, and have the track name so they can be loaded into UCSC. So now you can just file transfer these ones down and upload them straight into UCSC. And I have all the class data loaded into UCSC in a session that I will give everyone access to. Yeah, we'll go from there. All right, so next step is visualizing the data. But what we've done today is started to analyze how many peaks did we have in our start, how many were in our intersecting, and then how do we get these files exported that we've just created in a way that we can then visualize on UCSC or IGB. And so we will tackle that next. And in the meantime, thank you, and be well.